জাহাঙ্গীরনগর বিশ্ববিদ্যালয়ে রিসার্চ এক্সিলেন্স হিসাবে প্রতিষ্ঠা করার জন্য গড়ে তোলা হয়েছে ওয়াজেদ মিয়া বিজ্ঞান গবেষণা কেন্দ্র। এই গবেষণা কেন্দ্রে শুধুমাত্র জাহাঙ্গীরনগর বিশ্ববিদ্যালয়ের শিক্ষক গবেষকগণই গবেষণা করেন না, দেশের অন্যান্য প্রতিষ্ঠানের যারা গবেষক আছেন তারাও গবেষণা করতে পারেন এবং এই গবেষণা কেন্দ্রে বিজ্ঞানের অত্যাধুনিক যে ক্ষেত্রগুলি আছে সেগুলি নিয়ে গবেষণা করা হয়। আজ 5 সেপ্টেম্বর 2018 তারিখে এই গবেষণা কেন্দ্রে অনুষ্ঠিত হচ্ছে একটা আন্তর্জাতিক প্রশিক্ষণ কর্মশালা ডিএনএ বারকোডিং নিয়ে। আমরা এখন কথা বলছি এই ডিএনএ বারকোডিং গবেষণার অন্যতম উদ্যোক্তা আয়োজক ডক্টর মনার হোসেনের সঙ্গে। স্যার, ডিএনএ বারকোডিং গবেষণা করছেন আপনি এবং আপনার সঙ্গে আরও অনেকে আছেন এই টিমের সদস্য হিসাবে। আপনি অনুগ্রহ করে আমাদেরকে বলবেন এই সম্পর্কে।
ডিএনএ বারকোড অ্যাকচুয়ালি সারা বিশ্বে এখন খুবই একটা পপুলার টপিক অ্যাকচুয়ালি গবেষণা করার জন্য। সেক্ষেত্রে বাংলাদেশও পিছিয়ে নেই, আমরাও এগিয়ে যাচ্ছি এবং ধন্যবাদ বিশ্ববিদ্যালয় এবং আমাদের ইউজিসিকে যারা ফান্ড দিয়ে আমাদের ল্যাব করার জন্য সহযোগিতা করেছেন। একটু সারা বিশ্বে যেসব প্রাণী আছে, বিশেষ করে পোকামাকড়, উপকারী এবং অপকারী মানুষ, সবাই এদেরকে শনাক্ত করার জন্য একটা ডিএনএ সিকোয়েন্স ব্যবহার করা হয় এবং সেটাই হচ্ছে ডিএনএ বারকোড, তার একটা প্রজাতি শনাক্তকরণের জন্য। তো এই ক্ষেত্রে আমরা পোকামাকড় নিয়ে কাজ করছি, বিশেষ করে যেসব পোকামাকড় ক্ষতিকারক আমাদের ফসলের জন্য সেগুলোকে দমন করা বা সেগুলো কন্ট্রোল করা, আবার যারা আমাদের উপকার করে যেমন মৌমাছি, প্রজাপতি, এদেরকে সংখ্যা বৃদ্ধি করা, এই সব কিছু মিলে আসলে আমরা এই ডিএনএ বারকোড আমাদের ল্যাবের প্রতিষ্ঠা করার উদ্দেশ্য।
Recommendations very quickly. Departmental senior teachers of different departments of our university and other universities to be present here and giving us encouragement. This is valuable time to encourage us for the betterment of this. I respectfully welcome to my colleagues from other universities like Dhaka University, Rajshahi University, organizing under this. Our aim is to expose the facilities of these faculties to the students and researchers of Jahannagar and other universities of Bangladesh. We always encourage our extend. We will always encourage and extend our full cooperation and support for them. The researchers are always welcome to use the equipment and facilities of this laboratory for their research. Though we took a little more time to implement the project, it was because of the initial delay in the allocation of space for the laboratory. By this time, we have completed the sequencing of more than 200 species. Those sequences are submitted to the NCBI, the Science Center, for its current cooperation to highlight activities and achievements of this laboratory. Finally, I hope you all will enjoy the training workshop very much and have a good time. Thank you very much.
Was described, and now we are going to produce or support to prepare or to make the resource person on the particular field, as with molecular techniques and especially on the DNA barcoding. So now I am requesting Professor Sadia Ahmed to tell a very of your feelings actually today's seminar.
And our students will have exposure to new equipments, new facilities which we do not have in our department. So I hope Professor Jabbar and Professor Monar Hussain and Professor Asna will recruit as many students they can for the higher education, because if we do not construct a base for the student, so this is the stepping stone, so they can go abroad and learn more about it. So we all need, but from my experience, I did not know anything about mosquito when I did my PhD from the School of Hygiene and Tropical Medicine. But we are encouraging it. So we should, because teaching, education, there is a difference between, I always say that is a degree holder and educated person. So people can be degree holder but not educated. So for this reason, I am saying that we can start from our department, and also we could, in our faculty there are some departments already.
Some space, because there will be necessary sometime to do mistake. So come in the workshop again. Thank you very much for recalling not all our university teachers and students, that the only participants, whether from the Russian University, some of our colleagues, and from Dhaka University and from Chittagong University also, there are some representatives. So this makes us really very much good in spirit, actually, working some good job. In this stage, in this place, so I would like to request to our chairman, Department of Zoology, to tell something about this work. Please, Professor Dr. Monolith Khan.
This because such a specialized laboratory is unique to Bangladesh, because nowhere else we have such a well organized laboratory with so much equipment. So I thank the organizers. Special thanks to Dr. Senthil Kumar. He has come all the way from Mizoram, although Mizoram is a neighbor state to Bangladesh, but we don't have much exchange, so this can be a good beginning. Thank you all for joining. Thank you.
Exciting experience for me as well. I thank the Department of Zoology, Jahangir University, and also the organizers, Professor Abdul Jabbar and Professor Munwar Hussain, for their kindness in inviting me. And I would like to mention that today in India it is the Teachers' Day, 5th September. So from the morning I have been receiving a lot of messages and wishes. So I am also happy that today I am here, and then I am going to get the blessings of almost many of the senior teachers here and the students and the scholars. So it's a great occasion for me as well to be here. And like Sir was mentioning about the neighborhood of Mizoram and Bangladesh, yes, even the government of India is now harboring upon a policy called as a Look East policy, where they want the countries, the neighboring.
So I will request to enter your presentation for your presentation, please.
On to the first presentation on some basics of barcoding. So Professor Manwar was telling me yesterday that students from different departments, and even I had browsed through this Jahangir University, where I had seen a lot of life science departments like biochemistry, genetic engineering, zoology. So there is, and he also mentioned to me that there are students from diverse departments. So I thought of giving a very small basic introduction about what is DNA barcoding. So always please feel free to stop me if you have some questions or queries. You can, ask you can add on if I have missed out something. So let us keep the session interactive. So it's going to be, useful for all of us. So let's start with some basics. What we have already known, in most of the barcodes, like if we even happen to enter into a supermarket and we buy some product, we have a barcode of any product, we take it out. So the person just scans the barcode, and all the information regarding the manufacturing place, the price, the date, the expiry date, the date of manufacturing, and the contents, everything is stored within the barcode. Phylogenetics have come in place. So basically a barcode gene, it should have sufficient diversity within itself. It should not be strictly a replica, a Xerox copy of the same gene should not be Xerox in all different species. There has to be some variations that has led to speciation event as well, and also the changes in their biochemical or physiological role. So all those content has to be stored, and that is sufficient enough to differentiate all the species. Then thirdly, like nowadays we have next generation sequencing, so people even go for whole mitochondrial genome sequencing at a very low cost. But as such, the next generation sequencing is error prone. It is not, accurate more than 90. Not a molecular biologist or a biotechnologist or a genetic engineering person, but basically he was a field ecologist. After 25 years of extensive research in the field on birds, because of the difficulties they faced in the field to identify the bird species, so he had hit upon the barcode gene, cytochrome oxidase 1 gene, which could serve as a global identifying any stage of life. Third is, like, especially if you talk about mosquitoes, right? Hundreds of species of mosquitoes flying around, and very difficult to identify those mosquitoes. So barcoding will be very much useful to unmask the lookalikes. And it reduces the ambiguity in the sense that there will not be any conflict. between the taxonomies. Generally, it happens that taxonomists from different museums will have different opinions about the identification or characters which should be looked upon for identification of the species. So, but here a single base sequence, about 750 base pairs, should be good enough, and no one can conflict on that, or no one will have a second opinion on that. Many more reasons we can speed up the encyclopedia of life. People say that we have so far only discovered about 4 to 5% of the species that is actually existing in this world, in this ecosystem. So how do we, speed up and identify the organisms before it becomes extinct? So, and similarly, it democratizes the access. We barcode a insect and then or a species, and then we deposit it in XCBI, and it is accessed by anyone across the world. So sitting here, we can have sequences of organisms which are found in Africa or Europe, and then we can incorporate those data into our analysis to make more meaning about life. So that is what this gives. Secondly, also in museums, thousands and thousands of specimens preserved and stored, but yet unidentified. So that is where we can, go in for understanding the species as well. For identification of species. So again, it is not only for identification, but we can even try to find out their functional roles. Like for example, cytochrome oxidase 1 present in a tiger and present in a cow or present in a bird, how does it differ and what gives its physiological and metabolic variations, right? Can be found because cytochrome oxidase 1 is linked with the respiratory chain system. So there are many things that is coming up, especially even in the field of microbes. By just sequencing the 16S species, the standard barcode marker in microbial species, we can talk about the functional role of those microbial species in different environments. And what does it do? What metabolic activity is being done by those microbes, or the environment, what kind of activity does it hold good? So these are some more informations, we spoke about cytochrome oxidase 1 in animal species. In plants, especially because plant DNA is much more complex, even much more than human DNA. So there has not been a consensus on a single barcode gene, just like animal species. And again, the diversity of plants like angiosperms, gymnosperms, so they have different characters and different evolutionary significance. So people have tried upon different genes, like for example MATK, the maturase K gene present in the chloroplast, RBCL, ITS, tRNH. So many genes have been exploited or tried even in plants, and the people have different barcodes for different genera or different different phylum within the plants. For fungus, of course, ITS is still used as a good marker, and as I mentioned, the 16S in general for many of the microbes. So when we talk about animal DNA especially, so the whole mitochondrial genome is about 16,500 base pairs. About 37 genes are present in the mitochondria. So people now go in for next generation sequencing, and very beautiful pipelines exist in the bioinformatics arena where we feed in the data, and if we have some basic knowledge about phylogenetics, anyone can work with those softwares. So it's not difficult at all because the pipelines have all been automated. Very strong bioinformatics pipeline is available for microbial species and humans. For other systems, it is still under development, and maybe in the future we will be having it. So apart from CO1, there are many other genes, protein coding genes, which are involved in the NADPH pathways, ND1 to ND6, cytochrome B, ATP gene. So even ATP genes or cytochrome or ND genes or other mitochondrial genes are also used as barcode genes or biomarker genes even for human disease genetics. For example, the D-loop gene, the displacement loop gene, which is found on the top region of the mitochondria, that is where the DNA exists in the triple stranded form, right? Because that is the origin of replication of the mitochondria. So people use the D-loop gene as a very good barcode gene for detecting cancer incidence. Even before a person can get, cancer, 5 years before a person can get cancer, if his or her D-loop gene is sequenced, and if those mutations which are probably leading to carcinogenesis are identified in comparison to the healthy people, right, or in a population-wide study, the person can be alerted that look, you have mutations or polymorphisms in your D-loop gene that may probably lead to cancer, because already it has shown that in this population cancer patients post this kind of mutations or polymorphisms. So you need to be careful. You need to have a healthy lifestyle. So such kind of diagnosis. So early diagnosis. So barcode helps in many human research as well. So Taylor et al. 1991, where we say that ITS can be used for species and genera. Right? If we study a species and a genera, because ITS is internal transcriber space of DNA, they form the secondary structures. So not only for, if we sequence the ITS1 or ITS2 regions, not only for identification, but we can develop the secondary structures, the hairpin loops, the bends, and those structures can also be used as a marker for identification of the species, are very different from each other. So that itself is a standard for identification of the species. Right? Of biology or molecular biology, we need to analyze the gene first and then talk about the gene which we use as a marker. At what level will it be able to differentiate between a species and a subspecies? Yeah. For example, protein coding genes like cytochrome oxidase 1, even though it is in mitochondria, it cannot mutate so rapidly because it is a protein coding gene, right? So the protein will alter and the organism may die out. So mutations will not be so rapid compared with ribosomal gene, 16S gene, right? So the rate of evolution of the genes has to be taken into account. Yeah.